Okay, so protein is also called polypeptide, students. Why protein is called polypeptide? Because many peptide bonds are present, that's why protein is also called polypeptide. But what is meant by peptone? Peptone andre meaning it is made up of around 50 amino acids. 50 amino acids idendre adu peptone anta karitara. ಈಗ more than 50 amino acids idre that is called polypeptide anta karitara. Got it? So ide difference between the peptone and polypeptide. Yesterday class only I explained about how the peptide bond formation is occur between the amino acids. Remember students, but every polypeptide chain they are having how many ends are there? How many terminals are there? 2 terminals are there. One is called N terminal and last one is called C terminal. N terminal is represents what? First amino acid. C terminal is represents last amino acid. Always first amino acid carboxylic group can bind with the amino group of the second amino acid. Correct? First amino acid carboxylic group, second amino acid amino group madhya which bond is formation of peptide bond formation is occur. But in this peptide formation, come on, which one is removal? Water molecule is removal. That's why it is one of the dehydration process. Correct? So peptide bond formationally ribozyme enzyme is very helpful. Ribozyme is also called peptidyl transferase, another name. Peptidyl transferase enzyme is commonly helpful. Okay sir, ಇಲ್ಲಿವರೆಗೆ ಗೊತ್ತಾಯಿತು. ಮತ್ತು ಏನು ಸರ್, ಮತ್ತು ಪ್ರೈಮರಿ, ಸೆಕೆಂಡ್ರಿ, ಟೆರ್ಷರಿ, ಕ್ವಾಟರ್ನರಿ ಅಂತ 4 ಸ್ಟ್ರಕ್ಚರ್ಸ್ ಆರ್ ದೇರ್ ಇನ್ ದ ಪ್ರೋಟೀನ್. ಏನಿಲ್ಲ, ಜಸ್ಟ್ ಇದು mRNA. ಸೊ mRNA ಮೇಲೆ ರೈಬೋಸೋಮ್ಸ್ ಆರ್ ಕಂಪ್ಲೀಟ್ಲಿ ಅಟ್ಯಾಚ್ಡ್. mRNA ಮೇಲೆ ರೈಬೋಸೋಮ್ಸ್ ಆರ್ ಕಂಪ್ಲೀಟ್ಲಿ ಅಟ್ಯಾಚ್ಡ್ ಲೈಕ್ ದಿಸ್ ಒನ್. ಕರೆಕ್ಟ್? ಸೊ if more ribosomes are present on mRNA, what we can call polyribosomes, or it is also called polysomes. But ribosomes function enandre they can helpful for synthesis of which one? Protein. Correct? Why? Because ribosomes are helpful for formation of the peptide bond between amino acid. Ashte kalsa, okay na? They are helpful for formation of the peptide bond between the amino acid. But who will bring that one, the amino acids? tRNA brings the amino acid. So tRNA is also, read that one, what is the genetic code on mRNA? Correct? mRNA ಮೇಲೆ ಏನು genetic code ಇದೆ ಅಂತ ತಿಳ್ಕೊಂಡು ಆ genetic code ಪ್ರಕಾರ only they can bring that one, the amino acids. ಅದಕ್ಕೆ in the molecular base of inheritance only we discussed all. Okay? 3 types of RNAs are there. mRNA function in the mRNA bring that one, the genetic information about specific protein. Correct? Specific protein genetic information they can bring. That's why it is called M for what? Messenger RNA. But genetic information is present in the form of codons. Correct? So codon meaning every 3 adjacent nucleotides. Every 3 adjacent nucleotides is, we can call them as one codon. But every codon is completely coded a specific amino acid. So that is about. Okay? But some codons matra it not specific any amino acid. Such type of codons what we can call nonsense codons, or it is also called stop codons, also called terminator codons ante karitara. Ee terminator codon function it will be stop that one, which is synthesis? Protein synthesis. Remember UAA, UAG, UGA, these 3 are called. Understood, right? Okay? So now this is about the protein. Okay na? So protein is completely synthesized here, students. Protein is synthesized. Protein ಅಂದ್ರೇನೆ it is one of the chain of amino acid ಅಂತ ಹೇಳ್ಬೋದು. All amino acids are completely bind with each other by peptide bond. Clear? That's why it is also called string of amino acid ಅಂದ್ರೆ ಏನು ಹೇಳ್ಬೋದು? Correct? String of amino acid. Which one is called string of amino acid? Protein. Correct? Protein. ಯಾಕಂದ್ರೆ so many bead like structures are there. Okay na? It is also string of amino acid ಅಂತ. But primary structure ಅಂದ್ರೆ ಈಗ just freshly synthesized protein ನೇ ನಾವು primary structure of protein ಅಂತ ಕರೀತೀರಾ. ಅರ್ಥ ಆಯ್ತಾ? Okay? So it is commonly it is arranged in that one. You see, this about the proteins and the amino acids and the tilkoda. So e amino acids all are completely arranged. Amino acids are completely arranged like one linear structure. Linear structure nalli irutara. Okay? So this is about the, okay? So in the linear structure, in this structure, this is called N terminal anta perfect agirutte, and this one also C terminal anta perfect agirutara. Okay? So that's why, what is the meaning of primary structure? Primary structure means what? It is a linear arrangement of amino acids in the which one? Protein. Plus idu folded. Primary structure andre it is not showing any foldings, it is the unfolded structure. Idu important. Unfolded structure of protein, is called primary structure anta helbodu. But the primary structure is commonly having two important functions are there. Ena two important functions andre, so how many amino acids are there anta tilkobeku andre primary structure help madutera. It is commonly helpful for the number of amino acids in the protein. Second, it is also helpful for arrangement of amino acids in the protein. Clear students? Okay na? So amino acids ಏನೇನಿದೆ plus how many number of amino acids ಇದೆ ಅಂತ ನಾವು ತಿಳ್ಕೋಬೇಕಂದ್ರೆ ನಾವು we have to analyze the primary structure of protein or secondary, tertiary or quaternary. Primary structure ಮಾತ್ರ analyze ಮಾಡಬೇಕು. No need analyze that one. ಯಾಕಂದ್ರೆ so secondary ಆಗ್ಬೋದು, tertiary structure ಆಗ್ಬೋದು, they are completely change their shape. Understood? ಅದಕ್ಕೆ primary structure is very helpful in the two functions I already said. What are the two functions? It is helpful for analysis of number of amino acids. Second, it is also helpful for arrangement of amino acids. ಏನಿದೆ ಅಂತ ನಾವು primary structure ಇಂದನೇ ಗೊತ್ತಾಗುತ್ತೆ. But primary structure of the protein is unstable. ಅಂದರೆ it is not more time it is present. It is not even present in more time. Immediately primary structure is become which one? The secondary structure ಅಂತ ಕರೀತಾರೆ. ಸ್ಟೂಡೆಂಟ್ಸ್ ಅರ್ಥ ಆಗ್ತಿದೆಯಾ ಸ್ಟೂಡೆಂಟ್ಸ್? ಓಕೆ, ಏನಿಲ್ಲಪ್ಪ, ಜಸ್ಟ್ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಆಯ್ತಪ್ಪ ರೈಬೋಸೋಮ್ಸಿಂದ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಆಮೇಲೆ ಆ ಪ್ರೋಟೀನ್ ಹೆಂಗಿರುತ್ತೆ ಅಂದರೆ ಲೀನಿಯರ್ ಆಗಿರುತ್ತಲ್ಲ. ಅದಕ್ಕೆ ಲೀನಿಯರ್ ಅರೇಂಜ್ಮೆಂಟ್ ಆಫ್ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಇನ್ ದ ಪ್ರೋಟೀನ್ ಚೈನ್ ಆರ್ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಚೈನ್ ಈಸ್ ಕಾಲ್ಡ್ ಪ್ರೈಮರಿ ಸ್ಟ್ರಕ್ಚರ್ ಅಂತ ಕರಿತೀರ. ಜಸ್ಟ್ ಫ್ರೆಶ್ ಆಗಿದೆ ಅಂತ ಹೇಳ್ಬೋದು. ಕ್ಲಿಯರಾ? ಓಕೆ, ಸೊ ಇಟ್ ಈಸ್ ಹೆಲ್ಪ್ಫುಲ್ ಫಾರ್ ಟು ಫಂಕ್ಷನ್ಸ್. ಈಗ ಸೆಕೆಂಡರಿ ಸ್ಟ್ರಕ್ಚರ್ ಬಗ್ಗೆ ಡಿಸ್ಕಸ್. ಓಕೆ, ಸೆಕೆಂಡರಿ ಸ್ಟ್ರಕ್ಚರ್ ಅಂದರೆ ಓಕೆ ದ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಚೈನ್. ಓಕೆನಾ? ಸೊ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಚೈನ್ ಈಸ್ folded to form helix. Is called secondary structure. Polypeptide chain is folded to form helix. Is called them as secondary structure anta kariತಾರೆ. Secondary structure. ಏನು ಸರ್ secondary structure? Just before I said primary structure is over. But are you remember students polypeptide structure? Polypeptide structure ನಲ್ಲಿ every nitrogen base, okay? Every amino acid, how it is looks like, just I'm writing now. Here NH2 is commonly present. Next one, this is called CHCO. You go here only, which bond is there? Yes, peptide bond is there. Matte NH is there. Next one CH, matte CO. And dear students, matte, okay? Yes. Matte which one is there? NH and CH and CO. Like this one, it is present. You go ide. This is what we can call a yes peptide bond anta ಕರೀತಾರೆ. Clear? This is called peptide bond ಅಂತ ಹೇಳ್ಬೋದು. ಈಗ ನಾವು ಸೆಕೆಂಡರಿ ಸ್ಟ್ರಕ್ಚರ್ ಅಂದರೆ ಈ ಲೀನಿಯರ್ ಶೇಪ್ಡ್ ಪ್ರೋಟೀನ್ ಇಟ್ ಈಸ್ ಬಿಕಮ್ ಕಾಯಿಲ್ಡ್ ಲೈಕ್ ದಿಸ್. Why it is become coiled means, you see, here C double bond O is there, here NH is there. Clear? In single polypeptide chain, what is happened means between the oxygen and hydrogen, one of the hydrogen bonds formation is occurs. Because of the formation, you see, suppose here C double bond O is there. Next one is there, here NH is there. Between this one, what will be happening? Hydrogen bond formation is occurs. Hydrogen bond yava formation auto, again what will be? And these proteins or these amino acids, they will come close like this. When they will come close, what is happened? Linear shaped polypeptide chain, it is become which one? Helical shape. Again they should have N terminal is there and C terminal as well is there. So like this, it is, if it is present like this one, hydrogen bonds are present. Okay na? The hydrogen bonds idre, that is what we can call secondary structure anta ಕರೀತಾರೆ. ಈ secondary structure is present in the form of, okay, secondary structure present in present in alpha helix, and it is also called beta plate sheet anta karitar. Beta plate sheet. So alpha helix is completely formed by intermolecular bond, intermolecular hydrogen bond, sorry, intramolecular hydrogen bond. Okay, and beta plate sheet structure formed by, it is completely formed by intermolecular hydrogen bond. Intermolecular hydrogen bond. Clear students? Secondary structure nalli two format we can observe on alpha helix and beta plate sheet. Two parts are we can observe that. Okay, now you have to speak out. This is secondary structure. This secondary structure nalli it is alpha helix or beta plate sheet? Alpha helix only. Why? Because intra means what? Inside the polypeptide chain only hydrogen bond formation is occurs. That's why what will be happened? Okay, so it is become helical. But the second type is there, but it is not there in your NCERT. That is called beta plated sheets. This beta plated sheets, how it is looks like means what? You see, this about the one polypeptide chain and one more polypeptide chain also there. This polypeptide chain between hydrogen bond formation is occur like this one. This hydrogen bond formation is a completely beta plated anta karitira. Clear friends? Okay, so this is called beta plated sheet. Is a structure is formed by intermolecular hydrogen bond. Intermolecular, inter andre in between. So in between the polypeptide chain, if the hydrogen bond is formed means, okay, that is called beta plated sheet anta karithivi. Ee beta plated sheets we can observe in the example is keratin. Keratin is the best example for that one. Keratin is the best example for secondary structure of the protein. Yeah, best example keratin. You already know ee keratin where it is present. Hair nal irutte, nails nal irutte, correct? Even where it is also present, even our skin also. Okay, now skin also covered by keratin layer is present. Okay, it is a non-cellular keratin layer is present, students, above the skin. It is also acts as a which barrier? Physical barrier anta karitira. Physical barrier keratin. So that is about idu ella secondary structure nalli we can observe. The third one is called the mass tertiary structure. Third one is called tertiary structure, students. Tertiary structure. Tertiary structure. Okay, and very important secondary structure nodavaga. In secondary structure, okay, in proteins only right-handed helix are present. Right-handed helix are present. Mainly right-handed helix only present. Right side helix ಆಗಿರುತ್ತೆ. Right side helix ಆಗಿರುತ್ತೆ. So that's why it looks like a staircase ತರ ಇರುತ್ತೆ. It will be looks like a staircase ತರ ಇರುತ್ತೆ. So that is about. ಈ staircase ಹೆಂಗೆ ಅಂದರೆ suppose if you are going to that one that is about ಇದೆ ಮಾಲ್ಸ್ನಲ್ಲಿ ನೋಡಿದಿರಾ? ಓಕೆನಾ? ಸೊ ರೊಟೇಟ್ ಆಗಿರುತ್ತೆ ಗೊತ್ತಾ? ಸ್ಟೇರ್ ಕೇಸ್ ಇಟ್ ವಿಲ್ ಬಿ ರೊಟೇಟ್. ಅಂದರೆ ವೆನ್ ಯು ಆರ್ ಎಂಟ್ರಿಂಗ್ ಟು ದ ಮಾಲ್, ಎಂಟ್ರೆನ್ಸ್ ಡೋರ್ ಇಸ್ ದೇರ್, ಈ ಎಂಟ್ರೆನ್ಸ್ ಡೋರ್ ಸಮ್ ಆಫ್ ದಿ ಏರಿಯಾಸ್ನಲ್ಲಿ ಈ ಥರ ರೊಟೇಟ್ ಆಗಿರ್ತೀರ. ನಾವು ಹೋಗಿದ ತಕ್ಷಣ ಏನಾಗುತ್ತೆ ಅಂದರೆ ಓಪನ್ ಆಗುತ್ತೆ, ಮತ್ತು ಆಟೋಮೆಟಿಕಲಿ ಅದು ಕ್ಲೋಸ್ ಆಗುತ್ತೆ. ಹಿಂಗೆ ಬಂದು ಹಿಂಗೆ ಹೋಗೋದಲ್ಲ, ಓಕೆನಾ? ಅದೇ ರೊಟೇಟ್ ಆಗುತ್ತೆ ಗೊತ್ತಾ? ಸೊ ಆ ಥರ ಇದ್ದರೆ ದಟ್ ಈಸ್ ವಾಟ್ ವಿ ಕ್ಯಾನ್ ಕಾಲ್. ಸೊ ಪ್ರೋಟೀನ್ ಆಲ್ಸೋ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದ ಫಾರ್ಮ್ ಆಫ್ spherical or mainly helical staircase. It looks like a helical staircase. That is what we can call a secondary. Mainly in proteins only right-handed helix are majorly present. Right-handed, but okay, in other places of protein, okay. So and other direction is also present. Other places of protein also, other direction is also present. Andre majorityly right-handed direction irutte, which is structure secondary structure, but in some cases it is present ಲೆಫ್ಟ್ ಹ್ಯಾಂಡ್ ಡೈರೆಕ್ಷನ್ ಆಲ್ಸೋ ಇರ್ಬೋದು, ಲೆಫ್ಟ್ ಹ್ಯಾಂಡ್ ಡೈರೆಕ್ಷನ್ ಆಲ್ಸೋ ಇರ್ಬೋದು. ಬಟ್ ಮೆಜಾರಿಟಿ ಮಾತ್ರ ಇಟ್ ಈಸ್ ದೇರ್ ಎನ್ ಸಿ ಆರ್ ಟಿನಲ್ಲಿ ಸ್ಪೆಸಿಫಿಕ್ ಆಗಿ ಮೆನ್ಷನ್ ಮಾಡಿದರು. ಮೆಜಾರಿಟಿಲಿ ಸೆಕೆಂಡರಿ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ದಿ ಪ್ರೋಟೀನ್ಸ್ ಆರ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ರೈಟ್ ಹ್ಯಾಂಡೆಡ್ ಆರ್ ಲೆಫ್ಟ್ ಹ್ಯಾಂಡೆಡ್? ರೈಟ್ ಹ್ಯಾಂಡೆಡ್. Got the clarity? Okay, third one we can go for tertiary structure. Tertiary structure, I already written here. What is meant by tertiary structure? So tertiary structure andre meaning, okay? So here, what is the meaning of tertiary structure? So here the protein thread folded more and more, okay, to form woolen ball like to form woolen ball is called tertiary structure. It looks like a woolen ball. And nodi already helical shaped idella. Ee helical shaped, it will be showing that one so much of foldings are like this. Idralli also matte N terminal is there, C terminal is there. Students, more folding is present. If the more foldings are present means that is what we can call tertiary structure. This more folding main reason is same hydrogen bonds are there. Okay, na? Reasons are, what is the reasons for? Okay, first of all same more hydrogen bonds formation plus disulfide bridges formation ಆಗಬಹುದು. Okay, na? Next one, ionic bonds ಆಗ್ಬೋದು. ಇವೆಲ್ಲ ಫಾರ್ಮೇಷನ್ ಆಗಿ ಗೊತ್ತರ between the amino acids disulfide bridges and ionic bonds is completely formed, you know. ಈ ಥರ ಫಾರ್ಮ್ ಆವಾಗ ಏನಾಗುತ್ತೆ ಅಂದರೆ ಈ ಹೆಲಿಕಲ್ ಶೇಪ್ಡ್ ಹಿಂಗೆ ಹೆಲಿಕಲ್ ಆಗುತ್ತರ, ಹಿಂಗೆ ತುಂಬ ಮೋರ್ ಕಾಯಿಲ್ ಆಗಿ ಇಟ್ ಲುಕ್ಸ್ ಲೈಕ್ ಎ ವುಲನ್ ಬಾಲ್ ಹೆಂಗಿರುತ್ತೋ ಆ ಥರ ಪ್ರೋಟೀನ್ ಕನ್ವರ್ಟ್ ಆಗುತ್ತಾರೆ. ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಟೆರ್ಷರಿ ಸ್ಟ್ರಕ್ಚರ್ ಅಂತ ಕರಿತೇವೆ. ಆ್ಯಂಡ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. Okay, and it gives 3D structure of protein. This is very important. I should highlight, students. ಯಾವಾಗ ಪ್ರೋಟೀನ್ ಸ್ಟ್ರಕ್ಚರಿಗೆ ಒಂದು 3 dimension ಸ್ಟ್ರಕ್ಚರ್ ಬರುತ್ತಂದರೆ ಬಿಕಾಸ್ ವೆನ್ ದೇ ಆರ್ ಎಂಟರ್ ಇನ್ ಟು ದ ವಿಚ್ ಸ್ಟೇಜ್ ಟೆರ್ಷರಿ ಸ್ಟ್ರಕ್ಚರ್. ಓಕೆನಾ? ಆ್ಯಂಡ್ it gives that one biological importance of proteins. Okay, na? It gives biological importance of proteins. So very important, friends. Tertiary structure ge protein convert ಆಯಿತಂದ್ರೆ yes, ಆ proteins are modified into enzymes ಅಂತ ಅರ್ಥ. ಅಂದರೆ those are converted or those are enter into the tertiary structure ಅಂದರೆ they are enter into, okay, enzymes. ಅದಕ್ಕೆ mainly all enzymes are protein ಅಂತ ನಿನ್ನೆ ಹೇಳಿದ್ದೀರಾ. ಅಂದರೆ all enzymes are proteins but all enzymes are commonly present in the which structure? Tertiary structure only. Very important. ಅದಕ್ಕೆ tertiary structure ಬರೆಯುವಾಗ ಓಕೆನಾ. ಸೊ ಈ ಸೇ tertiary structure ಬರ್ತದೆ ಅಂದ್ರೆ you see this about the one of the tertiary structure ಅಂತ ತಿಳ್ಕೊಬೋದ್ರಾ. ನೋಡಿ ಈ tertiary structure ನಲ್ಲಿ ಸ್ಟೂಡೆಂಟ್ಸ್ ಆರ್ ಯು ಅಬ್ಸರ್ವಿಂಗ್ ಇಟ್ ವಿಲ್ ಬಿ ಫಾರ್ಮ್ ಸ್ಮಾಲ್ ಕ್ರೆವಿಸಸ್ ಅಂತ ಕರೀತೀರಾ. Tertiary structure nalli are observing that one protein is showing more coiling. Because of the more coiling it looks like a woolen ball and they are leaving a small crevices. Igo ee crevices ne ನಾವು ಏನಂತ ಕರೀತೀರಾ? Active sites ಅಂತ ಕರೀತೀರಾ. Active sites or it is also called binding sites ಅಂತ ಕರೀತೀರಾ. Binding sites. Igo ee binding sites are specific for every enzyme. Binding sites are specific for every enzyme. So one enzyme can be act on one substrate only. Why? Because what is the active or binding sites are there, it is suitable for one substrate only. Correct? That's why enzyme specificity we can observe because of the which one the active sites are which one binding sites. So ಈ binding sites are nothing but in the crevices ಅಂದ್ರೆ ಏನರ cracks ಅಂತ ಕರೀತೀರಾ. ನಾವು ವಾಲ್ ಮೇಲೆ ಕ್ರ್ಯಾಕ್ಸ್ ಬರುತ್ತಲ್ವಾ, ಓಕೆನಾ. ಸೊ ಅದೇ ತರಹ ಎಂಜೈಮ್ ಮೇಲೆ ಮೇನ್ಲಿ ವೆನ್ ದ ಪ್ರೋಟೀನ್ ಈಸ್ ಎಂಟರಿಂಗ್ ಟು ದ ಟೆರ್ಷರಿ ಸ್ಟೇಜ್ ಆರ್ ಟೆರ್ಷರಿ ಸ್ಟ್ರಕ್ಚರ್, ವೆನ್ ದ ಪ್ರೋಟೀನ್ ಈಸ್ ಎಂಟರಿಂಗ್ ಟು ದ ಟೆರ್ಷರಿ ಸ್ಟ್ರಕ್ಚರ್ ಇಟ್ ವಿಲ್ ಬಿ ಶೋಯಿಂಗ್ ದ ಕ್ರೆವಿಸಸ್. ಆ ಕ್ರೆವಿಸಸ್ಸೇ ಇಟ್ ಇಸ್ ಕಾಮನ್ಲಿ ಪ್ರೊವೈಡ್ ದಟ್ ಒನ್ ಬೈಂಡಿಂಗ್ ಸೈಟ್. ದಿಸ್ ಬೈಂಡಿಂಗ್ ಸೈಟ್ ಈಸ್ ಸ್ಪೆಸಿಫಿಕ್ ಫಾರ್ ಪರ್ಟಿಕ್ಯುಲರ್ substrate. Clear? So that's why, okay, So e point very very important. All enzymes are present in tertiary structure. This is very very important for your competitive exams. Ella enzymes are tertiary structure nalli iruttara, because enzymes are made up of protein, correct? So proteins are modified into enzyme andre compulsory protein should be enter into the tertiary. Okay, okay, next one more example also I'm completely telling na, ade myoglobin also one of the best example for a tertiary structure of the protein. Remember students, myoglobin where it is present? Muscles, correct? Red muscles are white muscles. Red muscles nalli myoglobin is there. But myoglobin is contain how many polypeptide chain? One polypeptide chain, correct? That's why one myoglobin can carries one oxygen only, not more than one oxygen. Why? Because they are having how many polypeptide chain? One poly. Okay, it is about the first discovered tertiary structure of protein. Very very important. Which one is the first tertiary structure of the protein? First discovered tertiary structure of the protein means what is the name? Myoglobin. Myoglobin is the first discovered tertiary structure of the protein. Protein tertiary nalli ide anta first ಎಲ್ಲಿ ಐಡೆಂಟಿಫೈ ಮಾಡೋದು ಅಂದರೆ ಮಯೋಗ್ಲೋಬಿನ್ನಲ್ಲಿ ಐಡೆಂಟಿಫೈ ಮಾಡ್ತಾರೆ. ಕ್ಲಿಯರ್ ಸ್ಟೂಡೆಂಟ್ಸ್? ಓಕೆ, ಸೊ ಪ್ರೈಮರಿ ಓವರ್, ಸೆಕೆಂಡರಿ ಓವರ್, ಟೆರ್ಷರಿ ಓವರ್. ಆರ್ ಯು ಅಬ್ಸರ್ವಿಂಗ್? ಗ್ರಾಜುವಲಿ primary structure convert into secondary convert into tertiary later convert into next one, last one, quaternary structure. That one, it is the fourth structure of the protein, quaternary structure. Got the clarity students? Okay na, tertiary structure anta gottaita? Okay, mainly all enzymes are present in the which one? Yes, tertiary structure only, because all enzymes are protein except ribozyme. All enzymes are proteins only, but enzymes are present in the tertiary structure. And which structure of the protein is give the 3-dimension structure? Tertiary only. Plus it is also give the biological importance. Andre ಒಂದು ಪ್ರೋಟೀನ್ ಒಂದು ಪರ್ಫೆಕ್ಟ್ ಕೆಲಸ ಮಾಡಬೇಕಂದ್ರೆ ಕಂಪಲ್ಸರಿ ದೇ ಶುಡ್ ಎಂಟರ್ ಇನ್ ಟು ದ ವಿಚ್ ಸ್ಟೇಜ್? ಟೆರ್ಷರಿ ಸ್ಟ್ರಕ್ಚರ್ಗೆ ಎಂಟರ್ ಆಗಬೇಕು. ಅವಾಗನೇ ಅದಕ್ಕೆ ಬಯಾಲಜಿಕಲ್ ಕೆಪಾಸಿಟಿ ಬರುತ್ತೆ, biological capability ಬರುತ್ತೆ. ಅದಕ್ಕೆ the biological importance of protein, it give that one, the biological importance of protein. ಈಗ let us we have to go for last structure of the protein, ಅದೇ quaternary structure. Quaternary structure. Okay, quaternary in the meaning that is called them as gathering or okay, yes, gathering of one, two or more than two, more than one polypeptide chains, more than one polypeptide chains or subunits, okay, arranged in different architecture arranged in different architecture such as cube or plate. Cube or plate is called quaternary structure. Quaternary structure ಅಂದ್ರೆ ಏನು? ಹಿಯರ್ ಮೋರ್ ದ್ಯಾನ್ ಒನ್ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಚೈನ್ಸ್ ಗ್ಯಾದರ್ ಆಗ್ತಾ ಇದೆ. more than one polypeptide or subunits, they are arranged in a different architecture. ಏನ different architecture? Either in the form of cube or either in the form of plate. In the form of plate or in the cube form, and they can gather. That is what they can form now quaternary structure ಅಂತ ಕರೀತಾರೆ. It is giving that one quaternary structures. Okay, it give that one the quaternary structure students. What is the best example for quaternary structure means? That is called hemoglobin is the best example for that. Hemoglobin. Clear students? Hemoglobin. Why? Because hemoglobin nalli 2 alpha chains and 2 beta chains are there. 2 alpha chains and 2 beta chains are there. The alpha chains and beta chains, nodra, more than one polypeptide chain anta kirtira. You see, this is about the one of the polypeptide chain anta tilkole. Okay, and it is also one of the polypeptide chain ಅಂತ ತಿಳ್ಕೊಳ್ಳಿ. ನೆಕ್ಸ್ಟ್ ಒನ್ ಮೋರ್ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಚೈನ್ ಇದೆ, ಈ ಥರ ಒನ್ ಮೋರ್ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಚೈನ್ ಇದೆ, ಇನ್ನೊಂದು ಪಾಲಿಪೆಪ್ಟೈಡ್ ಚೈನ್ ಆಲ್ಸೋ ದೇರ್. ಓಕೆನಾ, ಬಟ್ ಸೇಮ್ ಕಲರ್ ಆಲ್ಸೋ ಐ ಆಮ್ ರೈಟಿಂಗ್. ಎಸ್, ಇದೇ ಗ್ರೀನ್ ಕಲರ್ ಬರ್ತಾ ಇದೆ ಸ್ಟೂಡೆಂಟ್ಸ್. ಈ ಥರ one or more than one polypeptide chains, they can accumulate or gather. They will form that one particular either cube like structure or plate like structure is called quaternary structure. Best example is hemoglobin. Okay, you already know. Every alpha chain is consist of how many amino acid? How many amino acid in every 141 amino acids anta helidira. Alpha chain is consist of 141 amino acid. Beta chain is consist of 146 amino acids anta kelidira. Correcta? Okay, but hemoglobin nallira, are you observed students, heme and globin anta parts are present. Gota, heme is completely which part? Iron part anta kelidira. So nodi 4 irons are there. Correcta? 4 iron components are there plus, okay, yes, like this one. This is called alpha 1 chain, alpha 2 chain, beta 1 chain, beta 2 chain anta kelidira. Ide hemoglobin. This is adike one hemoglobin can carry how many oxygen? 4 oxygen. Why? Because they are having that one, the 4 polypeptide. But ade myoglobin also one type of hemoglobin, but it is about which structure? Tertiary structure. Tertiary structure andre only one polypeptide chain only present, but quaternary matra more than one polypeptide. Ide important for, okay, quaternary and tertiary structure ge ide main important students. Artha agutana? So this is complete. Total structure of the protein is finished. Remember friends? Okay, so this is about the protein structure, primary, secondary, tertiary and quaternary structure. But all enzymes are present in the which structure? Yes, tertiary structure only it is present. Okay, idra protein bagge complete information over, including protein functions also. Yesterday class we discussed functions of the protein also we discussed, but today class one more bio macromolecule, ade carbohydrates. Again, nucleic acid ಬಗ್ಗೆ ಮಾತ್ರ orally I can teach now. Why? Because nucleic acids you already familiar ಅಂತ ನಾನು ತಿಳ್ಕೊಂತಾ ಇದ್ದೀನಿ. ಕರೆಕ್ಟಾ ಸ್ಟೂಡೆಂಟ್ಸ್? Yes. Carbohydrates. What are carbohydrates? Carbohydrates andre this is called polyhydroxy aldehyde or ketones. Polyhydroxy aldehyde or ketones ni ನಾವು ಏನಂತ ಕರಿತೀರಾ? Yes, carbohydrates ಅಂತ ಕರಿತೀರಾ. ಈ carbohydrates, okay, so ಇದರಲ್ಲಿ 2 types of carbohydrates are there. This is called sugars and non-sugars. Sugars and non-sugars ಅಂತ 2 types are there. Sugars and non-sugars ಅಂತ 2 types are there. ಈ sugars say they are giving the energy ಅಂತ ಹೇಳ್ಬೋದು. ಈಗ next one, sugars are again divided into 2 types, that is called monosaccharides and second one is oligosaccharides. ಈಗ next one, yes, ಇದರಲ್ಲಿ only one type of only is there, that is called polysaccharides. At least carbohydrates also finished in your biomolecules. ಇದನ್ನು ಇಲ್ಲ ಅಂತ ಹೇಳ್ತೀರಾ? ಎಲ್ಲಿ monosaccharides ಅಂದರೆ ಇದು ಬೇಗ ಹೋಗುತ್ತೆ ಅಂತ ಅರ್ಥ. ಎಸ್, ಸೊ monosaccharides ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಸಿಂಪಲ್ ಶುಗರ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಸಿಂಪಲ್ ಶುಗರ್ಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಸಿಂಪಲ್ ಶುಗರ್ಸ್ ಯಾವುದು ಅಂದರೆ monosaccharides. simple sugars. And what is the formula of these monosaccharides? CnH2nOn anta karitara. CnH2nOn meaning ಏನು ಕಾರ್ಬನ್ ಹೈಡ್ರೋಜನ್ ಆಕ್ಸಿಜನ್ ಇಟ್ ಈಸ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ವಿಚ್ ರೇಷಿಯೋ 1:2:1 ರೇಷಿಯೋನಲ್ಲಿ ಇರುತ್ತೆ ಅಂತ ಅರ್ಥ. CnH2nOn. ಓಕೆ, ಈಗ ನೆಕ್ಸ್ಟ್ ಒನ್ monosaccharides. ಇಟ್ ಈಸ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ ಸಿಂಪಲ್ ಶುಗರ್ಸ್ ಅಂತ ಕರಿತೀರಾ. ಅಂದರೆ ವೆನ್ ಯು ಆರ್ ಈಟಿಂಗ್ ದ ಫುಡ್, when you are eating the food, we are eating in the form of which one? Polysaccharides or monosaccharide? Polysaccharide. Morning we are ate that one puliyogare. Puliyogare means what? That is totally what is the way? Starch is present. Correct? Starch after the eating, if they will enter into the stomach, 4 to 5 hours it will be is there in the stomach. After the stomach and this food is enter into which one? Small intestine. In which part of the small intestine complete digestion is finished? Duodenum only. But remaining jejunum and ileum part, which one is occurs? Absorption is occur, you already know. Here are students, okay? In the duodenum region only complete digestion is occur. Complete digestion is occur andre, what are the food we are taken? Polysaccharides, it will be converted into which one? Monosaccharides. Adike monosaccharides are called which one? Simple sugars anta kelidra. Simple sugars. Okay? Next, based on number of carbons, and monosaccharides are different types. Okay, monosaccharides are different types. The first one is called triosis, tetrosis, pentosis, hexosis, heptosis, and the different types are there, and heptosis. Well, different types are there, students. Ingene hogbodu. Heptosis ಆಮೇಲೆ octosis, nanosis, decosis ಅಂತಾನು ಹೇಳ್ಬೋದು. ಓಕೆ, ಟ್ರಯೋಸಿಸ್ ನಿಮ್ಗೆ ಗೊತ್ತು ಆಲ್ರೆಡಿ ಮೆನಿ OH ಗ್ರೂಪ್ಸ್ ಆರ್ ದೇರ್. ಅದಕ್ಕೆ ಪಾಲಿಹೈಡ್ರಾಕ್ಸಿ, ಪಾಲಿ ಮೀನ್ಸ್ ವಾಟ್? ಮೆನಿ, ಹೈಡ್ರಾಕ್ಸಿ ಮೀನ್ಸ್ OH ಗ್ರೂಪ್ಸ್ ಆರ್ ದೇರ್ ಪ್ಲಸ್ ಆಲ್ಡಿಹೈಡ್ ಆರ್ ಕೀಟೋನ್. ಐದರ್ CHO group is present or C double bond O group is there. C double bond O means what? Keto group. CHO group means what? Aldehyde group. So polyhydroxy, aldehyde or ketones we can call carbohydrates ಅಂತ ಕರಿ. ಮತ್ತು ಈ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ನಲ್ಲಿ monosaccharides when you are discussing first triosis. In this triosis, come on, how many carbons are there? 3 carbons are only present. ಅದಕ್ಕೆ it is called triosis. Can you give the example for triosis? Glyceraldehyde 3-phosphate is there plus DHAP is there. Remember students, glyceraldehyde 3-phosphate G3P ಅಂತ ಶಾರ್ಟ್ಕಟ್ ಆಗಿ ಬರ್ತಾ ಇದ್ದೀರಾ, ಪ್ಲಸ್ which one? DHAP also, dihydroxyacetone phosphate. Remember students, ಇದರಲ್ಲಿ which one is aldehyde, which one is keto? G3P is the aldehyde and DHAP is the keto group. But both are contain how many carbons? 3 carbons only. ಅದಕ್ಕೆ both are comes triosis ಅಂತ ಕರೀತಾರೆ. ಈಗ tetrosis ಅಂದ್ರೇನೆ 4 carbons are there. Can you give the example for tetrosis? That is called erythrose. Is just example. Erythrosis. It contain 4 carbons. ಈಗ next one, pentose ಅಂದ ತಕ್ಷಣ 5 carbons ಇದೆ ಅಂತ. Okay, for the example, ribose and ribulose also. Ribose and ribulose, it's the best example. Ribose sugar ಆಗ್ಬೋದು, even deoxyribose sugar also. Deoxyribose also, it contain how many carbons students? 5 carbons are present. 5 carbons are there, ribose and deoxyribose. Okay, ಇದು pentosis ಅಂತ ಕರೀತಾರೆ. ಈಗ ಲಾಸ್ಟ್ ಥರ್ಡ್ ಅಲ್ಲ, next one is called hexosis. Hexosis best example is glucose plus fructose. Glucose plus fructose is called them as hexosis. Okay, and pseudoheptalosis anta kettira. Pseudoheptalosis, it is generally calling as heptosis. 7 carbons, hepta anta takshana 7 carbons are there. Clear students? Later I can draw the diagram, I can give the atomic structure also. 5 carbons. Mainly we can discuss about, majority we can discuss about hexosis ಬಗ್ಗೆ majority we have to discuss. Okay? Okay, next one, why? Because pentosis ಬಗ್ಗೆ already we discussed in the nucleic acid. C5H10O4 ಇದ್ರೆ what we can call deoxyribose sugar. C5H10O5 ಇದ್ರೆ yes ribose sugar ಅಂತ. Okay? Okay, next one, oligosaccharides. Oligosaccharides also sugars ಅಂತ ಕರಿತೀವಿ, but it is made up of, okay na? So oligosaccharides is completely made up of 2 to 10 monosaccharide. 2 to 10 monosaccharides idre, that is what we can call oligosaccharides ಅಂತ ಕರಿತೀವಿ. ಅರ್ಥ ಆಯ್ತಾ ಸ್ಟೂಡೆಂಟ್ಸ್? ಓಕೆನಾ? ಇದೂ ಶುಗರ್ಸೇ, ಬಟ್ ಇದರಲ್ಲಿ monosaccharides 2 to 10 is present. And these oligosaccharides are again different types, no? That is called disaccharides, trisaccharides, okay na? Disaccharide, trisaccharide, tetrasaccharides, pentasaccharides, like different types are there. What is meant by disaccharide, students? Disaccharides meaning, okay na? Yes, two monosaccharides are, okay, bind, okay na? Bind with which one now? One bond is present. What is the name of the bond? Glycosidic bond. Understood? Between the sugars, which bond is there? Glycosidic bond. Very important. Glycosidic bond. So glycosidic bond is present in the oligosaccharides. Even it is also present in the polysaccharides also, students. And what is the best example for disaccharide? Disaccharides is best example in the sucrose. Next one, maltose. Next one, lactose. The mooru best example for this. Remember already digestion of sucrose. After the digestion of sucrose is converted into glucose plus fructose. Maltose, glucose plus glucose. Lactose, glucose plus galactose. That is about. ಈಗ ಟ್ರೈಸ್ಯಾಕರೈಡ್ಸ್ ಬೆಸ್ಟ್ ಎಕ್ಸಾಂಪಲ್ ಈಸ್ ಕಾಲ್ಡ್ ದ ಮಾಸ್ ಸ್ಟಾಕಿಯೋಸಿಸ್ ಅಂತ ಕರಿಬೋದು. ಸಾರಿ, ರಾಫಿನೋಸ್ ಇಸ್ ದ ಬೆಸ್ಟ್ ಎಕ್ಸಾಂಪಲ್ ದಾಟ್. ಇದರಲ್ಲಿ ಗ್ಲೂಕೋಸ್, ಫ್ರಕ್ಟೋಸ್, ಗ್ಯಾಲಾಕ್ಟೋಸ್ ಅಂತ 3 ಇರುತ್ತೆ ರಾಫಿನೋಸ್. ಈಗ ನೆಕ್ಸ್ಟ್ ಒನ್ ಸ್ಟಾಕಿಯೋಸಿಸ್. ಈಗ ಪೆಂಟಾ ಅಂತ ಹೇಳ್ಬೋದಾ, ವೆರ್ಬಾಸ್ಕೋಸಿಸ್ ಅಂತ ಕರಿತೀವಿ. ಓಕೆ, ಸ್ಟಾಕಿಯೋಸಿಸ್, ರಾಫಿನೋಸಿಸ್ ಇವೆಲ್ಲ ಬೆಸ್ಟ್ ಎಕ್ಸಾಂಪಲ್ ಫಾರ್. ಓಕೆ, ಟ್ರೈಸ್ಯಾಕರೈಡ್ಸ್ಗೆ ರಾಫಿನೋಸ್, ಟೆಟ್ರಾಸ್ಯಾಕರೈಡ್ಗೆ ಸ್ಟಾಕಿಯೋಸಿಸ್ ಇಸ್ ದ ಬೆಸ್ಟ್ ಎಕ್ಸಾಂಪಲ್. ಅಂದ್ರೆ ಗೊತ್ತಾಯ್ತಾರ? ಓಕೆ, ಟ್ರೈಸ್ಯಾಕರೈಡ್ಸ್ ಅಂದರೆ ಹೌ ಮೆನಿ monosaccharides are present? 3 monosaccharides are present. Students, how many are 3 monosaccharides present in the raffinose? Glucose, fructose, galactose. Galactose also present. Glucose, fructose, galactose. It is completely present in this one. Idaralli 4 monosaccharides are present. Clear? So this is oligosaccharides. Then we can go for polysaccharides. ಅರ್ಥ ಆಗ್ತಿರೋ ಸ್ಟೂಡೆಂಟ್ಸ್? ಓಕೆನಾ? ಇದು ಬೇಗ ಅರ್ಥ ಆಗುತ್ತೆ, ನೋ ಪ್ರಾಬ್ಲಮ್ ಅಲ್ವಾ ಗೌತಮ್? ಗೌತಮ್ ಅಂತ ಹೇಳ್ತೀರಾ? ಗೌತಮ್ ಅಂತ ಹೇಳ್ಬಾರ್ದು, ಗೌತಮ್. ನೆಕ್ಸ್ಟ್ ಒನ್, polysaccharides. Poly meaning what? Yes, many ಅಂದರೆ how many monosaccharides? Yes, more than 11. Very good. Or more than 10 monosaccharides. More than 10 monosaccharides binds to each other. Okay na? Each other by which bond? Glycosidic bond. Yes, bind to each other by glycosidic bond. Glycosidic bond. Same, that is polysaccharides. Again, polysaccharides also, how many types are there, students? 2 types of polysaccharides are there. Speak out what are the 2 types of polysaccharides. Homopolysaccharides and heteropolysaccharides. 2 types is there. It is very important. It is there in your textbook, students. Homopolysaccharides and heteropolysaccharides. Okay, can you speak out, students? What is the meaning of homo? Yes, same type of monosaccharides, correct? Okay na? So these are made up of, okay na? So this is completely made up of one or same kind of amino acid. Same kind of, okay, or same type of amino acid. Same type of amino acids is called homopolysaccharides. ಒಂದೇ ಒಂದು type of monosaccharides. Only one type of monosaccharides. The best example is there. Which one? Cellulose, glycogen, chitin. Cellulose, glycogen, chitin, inulin. Okay? The next one. Cellulose is a polymer of alpha glucose. All glucose molecules are alpha glucose. Glycogen, it is a polymer of beta glucose. beta glucose. ಈಗ chitin is just a glucose only polymer, but inulin is a polymer of very important of fructose. ಇದು ಇನ್ಸುಲಿನ್ ಅಲ್ಲಪ್ಪ ಸ್ಟೂಡೆಂಟ್ಸ್, ಇನ್ಯುಲಿನ್. ಮತ್ತು ಇನ್ಸುಲಿನ್ ಈಸ್ ಎ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಆರ್ ಪ್ರೋಟೀನ್? Protein. Insulin is a protein, but is inulin, it is a polymer of fructose that is called homopolysaccharides. Okay, cellulose, chitin, glycogen, inulin, these are all are the best example for homopolysaccharide. ಈಗ heteropolysaccharides ಅಂದ್ರೆ ಏನೇ ಮೀನಿಂಗ್? Hetero means what? Different monosaccharides are present. It is made up of different monosaccharides. It's made up of, okay, so it's made up of different monosaccharides. ಇದಕ್ಕೆ ಬೆಸ್ಟ್ ಎಕ್ಸಾಂಪಲ್ peptidoglycan is the best example, plus mucopeptides. ಇದು ಬ್ಯಾಕ್ಟೀರಿಯಾನಲ್ಲಿ ಇರುತ್ತಲ್ಲ, ಗೊತ್ತಾ? ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ ವಾಲ್, ಮ್ಯೂರಿನ್, mucopeptides, peptidoglycans, plus pectin also one type of heteropolysaccharide. Okay, then we can go for ಹೆಂಗೆ ಸರ್ ಇದು how these monosaccharides can be bind with each other. Okay, with the help of atomic structure I can explain now. Okay, so first of all, glucose structure I am writing now, student. Glucose structure already know, students. Glucose structure, when you have to this is about the glucose. Hello? Next one, fructose. This is about the fructose. But glucose, sometimes it is present in the which one? Circular form also it will be present. But when does it will be circular form means if yes, aldehyde group andre idu first carbon tane, aldehyde group if they can bind with the which one? Fifth carbon. Idu first, second, third, fourth, fifth, sixth tane, sixth carbon ಜೊತೆಗೆ bind ಆವಾಗ ಏನಾಗುತ್ತಂದ್ರೆ sometimes glucose become present in circular form. How it is looks? Clear? One water molecule will be gone. This one, oxygen is present because here OH, here hydrogen is there. Water molecule remove ali, one of the bond formation out there. That's why it is become circular. Same students, H, OH. Here also H, OH is there. OH, H, and this about the OH, H, and here CH2OH. Okay, no? First, second, third, fourth, fifth, sixth carbon. Clear, students? This is about the one of the glucose molecule. So glucose molecule, either it is present in the form of linear or either in the form of circular. ಈ glucose ನಲ್ಲಿ alpha glucose is there, beta glucose is there. Students, remember OH group, if it is present at the below, that is alpha. If it is above means what? Beta. But which one is the polymer of alpha glucose? Cellulose is a polymer of, okay, alpha glucose ಅಂತ ಕರೀತಾರೆ. ಅದಕ್ಕೆ ಸೆಲ್ಲೋಸ್ ಫಾರ್ಮೇಷನ್ ಆಗಬೇಕಂದ್ರೆ ಆಲ್ಫಾ 1,4 ಗ್ಲೈಕೋಸಿಡಿಕ್ ಬಾಂಡ್ಸ್ ಆರ್ ಪ್ರೆಸೆಂಟ್. ಹೆಂಗೆ ಅದು ಅಂತ ತಿಳ್ಕೋಣ. ಈಗ ಇದು ಅರ್ಥ ಆಯ್ತಲ್ಲ? ಈಗ ನೆಕ್ಸ್ಟ್ ಒನ್, ಗ್ಲೈಕೋಸಿಡಿಕ್ ಬಾಂಡ್, ಹೌ ದೇ ಕ್ಯಾನ್ ಫಾರ್ಮ್, ಜಸ್ಟ್ ಯು ಹ್ಯಾವ್ ಟು ಡಿಸ್ಕಸ್. ಗ್ಲೈಕೋಸಿಡಿಕ್ ಬಾಂಡ್ same here also. OH is present. ನೋಡ್ರಾ ಹಿಯರ್ ಆಲ್ಸೋ water molecule remove agi come on one bond is formed that is called glycosidic bond. ಈ glycosidic bond first glucose molecule first carbon, second glucose molecule fourth carbon alva. This is about the first, second, third, fourth. ಅದಕ್ಕೆ these linkages we can call them as 1,4 glycosidic bonds ಅಂತ ಕರೀತಾರೆ. 1,4 glycosidic bond, glycosidic linkage ಅಂತ ಕರೀತಾರೆ. ಸಪೋಸ್ ಆಲ್ಫಾ ಪೊಸಿಷನಲ್ಲಿ ಇದೆ ಅಂದರೆ ಆಲ್ಫಾ 1,4 ಗ್ಲೈಕೋಸಿಡಿಕ್ ಲಿಂಕೇಜಸ್ ಅಂತ ಕರಿತೀವಿ. ಬೀಟಾ ಇದ್ದಿರಿದ್ರೆ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಬೀಟಾ 1,4 ಲಿಂಕೇಜಸ್ ಅಂತ ಕರಿತೀವಿ. ಅರ್ಥ ಆಯ್ತಾ ಸ್ಟೂಡೆಂಟ್ಸ್? ಓಕೆನಾ? ಬೀಟಾ ಅಂದರೆ 1,4 ಅಂದರೆ ಫಸ್ಟ್ ಫಸ್ಟ್ ಗ್ಲೂಕೋಸ್ ಮಾಲಿಕ್ಯೂಲ್ ಫಸ್ಟ್ ಕಾರ್ಬನ್, ಸೆಕೆಂಡ್ ಗ್ಲೂಕೋಸ್ ಮಾಲಿಕ್ಯೂಲ್ ಫೋರ್ತ್ ಕಾರ್ಬನ್ ಮಧ್ಯ ವಿಚ್ ಬಾಂಡ್ ಈಸ್ ಫಾರ್ಮೇಷನ್ ಆಫ್ ಗ್ಲೈಕೋಸಿಡಿಕ್ ಬಾಂಡ್ ಫಾರ್ಮೇಷನ್ ಇಸ್ ಅಕರ್ಸ್. ಈ ಗ್ಲೈಕೋಸಿಡಿಕ್ ಬಾಂಡ್ ಫಾರ್ಮೇಷನಲ್ಲಿ ಡಿಹೈಡ್ರೇಷನ್ ಆಗುತ್ತೆ. Clear? One time ask her direct, what is the name of the bond is present between? What is the name of the bond is present between monosaccharides or polysaccharide or polysaccharide anta kelidra? Which one? Glycosidic bond. Clear? Okay, now I am drawing about one glycosidic structure. Okay, idu ಮತ್ತೆ repetition ಮಾಡಬೇಕಂತ ಬೇಕಾಗಿಲ್ಲ students, but in your examination they can ask that one. Which one is called fruit sugar anta keltara? Which one? Fructose is called the mass. Which one is called cane sugar anta keltara? Sucrose is called the mass. Okay, like that they can ask when small general questions they may ask students. Okay, then we can discuss about polysaccharides we can discuss that. One more important thing ra, ಈ ಹೋಮೋಪಾಲಿಸಾಕರೈಡ್ ಹೆಟ್ರೋಪಾಲಿಸಾಕರೈಡ್ ನೋಡ್ಬೋದು. ಓಕೆ, ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ಸ್ನಲ್ಲಿ ಮೇನ್ಲಿ ಪಾಲಿಸಾಕರೈಡ್ಸ್ ಐದರ್ ಹೋಮೋ ಆರ್ ಹೆಟ್ರೋಪಾಲಿಸಾಕರೈಡ್ಸ್. ಅದೇ ಪ್ರೋಟೀನ್ಸ್ ಆರ್ ಕಂಪ್ಲೀಟ್ಲಿ ವಿಚ್ ಒನ್? ಹೆಟ್ರೋಪಾಲಿಸಾಕರೈಡ್ಸ್. ಬಟ್ ದಿಸ್ ಒನ್ ಐದರ್ ಹೋಮೋಪಾಲಿಸಾಕರೈಡ್ಸ್ ಆರ್ ಹೆಟ್ರೋಪಾಲಿಸಾಕರೈಡ್. ವೈ? ಬಿಕಾಸ್ ಓಕೆ ಓನ್ಲಿ ಒನ್ ಕೈಂಡ್ ಆಫ್. ಅದಕ್ಕೆ ಸೆಲ್ಲೋಸ್ ನೋಡಿ, ಸೆಲ್ಲೋಸ್ ಇಸ್ ಎ ಪಾಲಿಮರ್ ಆಫ್ ವಿಚ್ ಒನ್? alpha glucose. Only alpha glucose molecules only present. Ella alpha glucose molecules nalli come on, alpha 1,4 linkages or beta 1,4 linkages alpha 1,4 linkages we can observe. Beta 1,4 linkages we can present in glycogen. Ee glycogen ಬಗ್ಗೆ ನೀವು ಹೇಳಬೇಕು. Glycogen is called which starch? Animal starch ಅಂತ ಕರೀತಾರೆ animals ನಲ್ಲಿ. ಓಕೆನಾ? ಇನ್ನೊಂದು ಪಾಯಿಂಟ್ ಆಲ್ಸೋ ಬಿಟ್ಟಿದೆ, starch also one of the best example for this. Starch also one of the homopolysaccharide ಅಂತ ಹೇಳ್ಬೋದು. starch, glycogen, cellulose, chitin, inulin, even starch also it is a polymer of the glucose only. But only one polysaccharide matra polymer of the fructose, name out inulin matra polymer of the fructose. Idu maybe they can ask the question for your competitive examinations. This one you should highlight on. Understood? Okay. Next, now I am completely drawing about one polysaccharide students. Okay. So I am drawing about the glycogen structure. I am drawing glycogen. Okay. ಈ glycogen ನೋಡಿದ್ರಾ? So ಈ ತರಹ it will be showing that one so many branches like this. It will be showing that one so many branches are present. Glycogen, it will be showing so many branches like this. So many branches nalli suppose E part. No, first E part. This is called right side anta karithivi. This is called left side anta karitira. Right side ne reducing part anta karitira gotta? Reducing end anta karitira. And this one is called non-reducing end anta karitira. Reducing end, non-reducing end anta. Two poles are present to polysaccharides. Reducing end anta ಯಾಕೆ ಕರಿತಾ ಇದ್ವಿ ಅಂದರೆ ಈ ರೀಜನ್ ನಲ್ಲಿ ಪಾಲಿಮರೈಸೇಷನ್ ವಿಲ್ ಬಿ ಕಂಟಿನ್ಯೂಡ್ ಟು ದಿಸ್ ಇಯರ್. ಅದಕ್ಕೆ reducing is occurs here. So polymerization we can't observe this side. Understood? That's why it is called non-reducing end ಅಂತ ಕರಿತೀವಿ. ಅರ್ಥ ಇದೆಯಾ? ಪ್ರೋಟೀನ್ಗೆ N ಟರ್ಮಿನಲ್ C ಟರ್ಮಿನಲ್ ಅಂತ ಇರುತ್ತಲ್ವಾ? ರಾ ಎನ್ ಟರ್ಮಿನಲ್ ಸಿ ಟರ್ಮಿನಲ್ ಅಂದರೆ ಎನ್ ಟರ್ಮಿನಲ್ ಯಾವ ಸೈಡ್ ಇರ್ಬೋದ್ರ ಲೆಫ್ಟ್ ಸೈಡ್ ಇಸ್ ದೇ ರೈಟ್ ಸೈಡ್ ವಿಚ್ ಒನ್ ಇಸ್ ದರ ಸಿ ಟರ್ಮಿನಲ್ ಸೇಮ್ ಹಿಯರ್ ಆಲ್ಸೋ ರೈಟ್ ಸೈಡ್ ವಿಚ್ ಒನ್ ರೆಡ್ಯೂಸಿಂಗ್ ಎಂಡ್ ಅಂತ ಕರಿತೀರ ಆ್ಯಂಡ್ ದಿಸ್ ಒನ್ ನಾನ್ ರೆಡ್ಯೂಸಿಂಗ್ ಎಂಡ್ ಅಂದರೆ ಈ ಸೈಡಿಂದನೇ ಓಕೆನಾ ಸೊ ವಿಚ್ ಒನ್ ಈಸ್ ಅಕರ್ಸ್ ಕಂಟಿನ್ಯೂಷನ್ ಆಫ್ polysaccharide continuation is occurs. Okay, now I can give that one how this now this part is there. ಈಗ ಈ ಪಾರ್ಟ್ ಎಲಾಬರೇಟ್ ಮಾಡಿ ನಾನು ಡ್ರಾ ಮಾಡ್ತಾ ಇದ್ದೀನಿ ಸ್ಟೂಡೆಂಟ್ಸ್. ನಾವು ದಿಸ್ ಇಸ್ ಅಬೌಟ್ ಒನ್ ಆಫ್ ದಿ ಗ್ಲೂಕೋಸ್ molecules already CH2OH is present students. Okay, next one HOH, OHH, and here also OHH is present, and here oxygen is present. Okay, then here also I already said totally ಈ ಮಧ್ಯ ವಾಟರ್ ಮಾಲಿಕ್ಯೂಲ್ ಹೋಗುತ್ತರ H2O ಇದರಲ್ಲಿ OH here OH ಹೋಗಿ ಸೊ ದಿಸ್ ಇಸ್ ಕಾಲ್ ವಿಚ್ ಬಾಂಡ್ ಗ್ಲೈಕೋಸಿಡಿಕ್ ಬಾಂಡ್ ಅಲ್ವಾ. ಇಲ್ಲಿ ವಾಟರ್ ಮಾಲಿಕ್ಯೂಲ್ ಹೋಗಿದ ತಕ್ಷಣ ಇಲ್ಲಿ ಆಕ್ಸಿಜನ್ ಹಂಗೆ ಇರುತ್ತಲ್ಲ ಸೊ ಅದೇ ಆಕ್ಸಿಜನ್ ಐ ರಿಟರ್ನ್ ಗಾಟ್ ಇಟ್. ಸೊ ಹಿಯರ್ ದಿಸ್ ಇಸ್ ಅಬೌಟ್ ದ CH2OH. Okay, like this we can observe here. This is called reducing end, this one is called non-reducing end, this is called reducing. But branches part nalli hengirutte, branches part, ee branches part how it will be looks like means small diagram I am drawing students. Okay, so here CH2OH ಪ್ಲೇಸ್ ಸಾಕಾಗ್ತಿಲ್ಲಪ್ಪ. ಅರ್ಥ ಆಯ್ತಾ? So many branches also present. We are discussing what about this structure of glycogen. ಇದು glycogen. ಅರ್ಥ ಆಯ್ತಾ? Glycogen is about the glycogen structure. Glycogen showing so many branches. ಈ branches ನಾವು ತಿಳ್ಕೊಂಡವಾಗ ಇದು ಲೀನಿಯರ್ ಸ್ಟ್ರಕ್ಚರ್ ಆಗಿ ಹಿಂಗೆ ನೋಡ್ತಾರೆ ಈ ಥರ. ಓಕೆ, ಮತ್ತೆ ಆಕ್ಸಿಜನ್ is there, it is completely bind with. But branching structure nalli CH2OH is there, you know, CH2OH it will be completely undergoes glycosidic bond with one more glucose molecule like this one more glucose molecule jotige, okay na, they can completely bind like this one, they can form, you see this one, it is indicates like this. Aita artha aita. Yes. Okay, next one, which polysaccharide they are giving blue color in iodine test? Starch. Why starch is giving that one blue color in the iodine test? Andre iodine andre inna, that is about I2. ಈ iodine easily captured in the helices, andre in the branches of the starch. Even starch also more or less looks like this one. Inga starch nalli gottara inga tumba iruttara. Okay na? Starch structure nodavaga ee tara branches, so many branches are present like this one. Ee starch, ee branches madhya iodine ella sikkutte. Means what, okay na? It will be completely trapped. If they can trap inside that one, inside the starch molecule, then they can give that one the which color? That is called blue color. Adike iodine will be trapped inside that one, the branches of the starch. Ade cellulose nodi. Cellulose is not showing branches. Cellulose is commonly showing like this. Parallel ಆಗಿರುತ್ತಲ್ಲ. All the so many glucose molecules are present like this parallel ಆಗಿ glucose molecules are present. ಇದನ್ನು homo polymer ಏನೋ. But so many linearly arranged, but it not showing any branches. Where does we can't show the branches wrong? Branches we can't observe in which one? Cellulose. ಅದಕ್ಕೆ cellulose ನಾವು iodine ಹಾಕಿದಂದ್ರೆ cellulose is not turned into the blue color. Why? Because they can't trap the iodine particles in there. Okay, in that structure, but here branches are there. E branches nalli they can trap that one. That's why starch is giving the blue color. So blue color kottide andre yes. The presence of starch or absence of the starch? Presence of the starch. Understood, student? Idra today class. Okay, this much is enough.